Hi everyone, and welcome to the Chapter 11 lecture for Chem 100. This is usually the last chapter I cover in the class, and you know the saying, they save the best for last? It's kind of how I feel about this chapter because I am a molecular biologist by training, so the molecules surrounding life, including DNA, are pretty near and dear to my heart. It's a long chapter, it's a very information dense chapter, and is often kind of challenging for students. And since we throw it in at the end of the class, you usually don't get to spend as much time with it as I wish we could spend. I could spend a whole course teaching this material. But so I try to give you all the information that you need to know before moving on to other biology classes where you may need to know this information. But I don't test you. There's no test on this. I mean, it will be on the final exam. But it's not covered on a unit test. So you only have to be able to survive the questions on the final exam. So don't panic that it's so much information. Just try to take in what you can, and hopefully you find it interesting and you learn something. All right, so this chapter is all about nucleic acids, DNA, and RNA. And these are really important molecules. They are basically the code that codes for how we are built and how we function. And here's my little meme. We'll talk about how one of the differences between DNA and RNA is that DNA is double-stranded and RNA is single-stranded. So DNA stands for deoxyribonucleic acid. It's a mouthful, but it describes the chemistry of what it's made of. It's a nucleic acid because it's found in the nucleus. It has acidic properties chemistry-wise. And we'll see the sugar, deoxy, is a ribose that's missing an oxygen. So it's called deoxyribonucleic acid. It's found in all of our cells, in the nucleus of the cell. In bacteria that don't have a nucleus, it's just found in the cell, in the cytoplasm there. But in human cells, and that's what we'll stick with in this Chemistry for Life class, it's found in the nucleus, and all of the DNA in the nucleus together collectively is referred to as our genome. Every cell in your body has the same DNA, every single cell. But your cells have but each cell looks different. So like a brain cell looks different than a skin cell, which looks different than a white blood cell. So there's tons of different types of cells in your body, but they all come from the same DNA. They all have basically the same instructions in them, but they're not all made the same way. So it's kind of like if you and 10 other people all had the same recipe book and you decided to make dinner using the same recipe book. You might have all made completely different dinners because you picked a different page in the recipe book. So just because you have all the same instructions doesn't mean you use all the same instructions. And that's kind of like how our cells are. They all have the same recipes at their disposal, but each one will make their own, decide which genes they want to express, or they're programmed to express or make certain genes into proteins. And that's why we have all these different cell types, even though they all have the same DNA, the same instructions. So let's look at the chemistry, the molecular structure of nucleic acids. So the building blocks of nucleic acids are called nucleotides. We're going to see that nucleic acids are polymers. They're long chains of repeating units, just like polysaccharides and polypeptides. And just like those things had individual unit building blocks, so do our nucleic acids. So nucleotides actually each nucleotide is composed of 3 parts. And there's a few different types of there's 5 different nucleotides. There's cytosine, thymine, guanine, adenine, and uracil. And these groups, these nitrogenous bases is what we call them, are attached to a sugar, a 5-carbon sugar, and attached to that is a phosphate group. So all nucleotides have 3 parts. They have a phosphate, a 5-carbon sugar, either ribose for RNA or deoxyribose for DNA. Notice that the deoxyribose is missing an oxygen at carbon 2 here. That's why it's called deoxyribose. And then one of these nitrogenous bases. Notice that the 3 on top all have a single ring structure, and they're known as the pyrimidines. And the 2 on bottom have a double ring structure and are known as the purines. So when we form nucleotides are formed by our very familiar synthesis reaction, which is condensation. So 2 things come together and water is produced. So first the base and the sugar get together, and then the phosphate joins in. And so in the end, you have a full nucleotide. So a nucleotide, this is an example of a nucleotide here. It has a phosphate group. It has a sugar, this one in this case is ribose, and it has a nitrogenous base. This one is adenine, which is a purine. It has 2 rings. Just a little terminology thing to point out here, the difference between nucleoside and nucleotide. Nucleoside is what we call the sugar and base before the phosphate is added, and nucleotide is what it's called after it's added. In the past, students in the homework would have questions that would ask about nucleosides versus nucleotides. I don't really care. You don't need to know that level of detail in this chapter. We're really going to just focus on nucleotides. Those are the complete unit of the building blocks for DNA. So now we can take these nucleotides and link them together in long chains to make DNA and RNA. Just a little bit about the naming of these different nucleotides. So if it's RNA, then we call it so adenine, the nucleotide for adenine is adenosine monophosphate, or AMP. And then for guanine, it's GMP, and so on and so forth. If we're talking about a DNA nucleotide, we put a little D in front of it to indicate the different sugar. So this first one in this little concept check down here, deoxyguanosine 5 monophosphate, DGMP, is DNA, whereas adenosine 5 monophosphate, or AMP, would be a nucleotide of RNA. AMP, adenosine monophosphate, is the precursor to ADP, adenosine diphosphate, which is the precursor to, ding, 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 ATP, our energy molecule. So that energy molecule that we create and use so much in the cell is actually a derivative or derived from nucleic acids, from an RNA nucleic acid. All right, so when we are forming these polymers, again, we're going to be performing condensation reactions. It's a classic synthesis reaction where we combine 2 things together, we lose water in the process. So an oxygen and 2 hydrogens are removed, and we end up with water, and these 2 things become linked together. This bond that holds them together is called a phosphodiester bond because of the phosphate and the 2 esters, which are these oxygens. So phosphodiester bonds link nucleotides together. You'll also see in the chapter, as we continue talking, that we'll talk about the reading frame for DNA and RNA, that it's a code, and we have deciphered the code. It's a code that's made up of 4 letters. And just like with proteins, we always read from N to C. We always read English from left to right, right? We always read nucleic acid sequences from the 5' end to the 3' end. And the 5 and 3, that just is talking about if you number the carbons in this ring, this carbon is number 5. If you number the carbons in this ring, this carbon is number 3. So the 5' end always has a phosphate sticking off of it, and the 3' end always has this alcohol group sticking off of it. I won't make you identify in a molecule which is the 5' and 3' end, but you do need to understand that when I say 5' to 3', I'm talking about the specific direction that we read the DNA in, just like we read English left to right, and we read protein sequences N to C, from the N terminus to the C terminus. It's also how nucleic acids are, how nucleotides are linked together. They're linked from 5' to 3' end. The 5' and 3' ends are where the phosphodiester bond forms. So when we talk about the nucleic acid sequence, I said we read it from 5' to 3'. So this sequence would be read GTCA, not ACTG. It would be read from the 5' to 3'. So I like to give a little picture of history when it comes to DNA, because I think we are so familiar with it now, even in, grade school, in cartoons that my daughter watches, they talk about DNA. It's in sci-fi. It's really kind of ubiquitous, the knowledge of DNA now. But it's really a very recent history, a very recent discovery in science history. So let's go back 300 or so years to 1750. And in 1750, DNA was not a word. It was not even fathomable to people. They understood a little bit about inheritance. They understood, they could observe that offspring often resembled their parents. That was a very observable phenomenon that they called inheritance. How parents pass traits onto their children was not understood at all. It was very magical, actually. And the reigning idea of the time, and this is a picture from a medical textbook of the time, was of a sperm. They literally thought that there was a teeny tiny little man inside the sperm, like a microscopic little man, that implanted itself in the womb and then grew into a baby. That's how they thought reproduction. They really didn't understand reproduction at all. And they thought that the homunculus, carried the traits of the father, but like magically absorbed the traits of the mother or something. There was just a lot of mysticism sort of surrounding inheritance. And in 1857, so this is 100 years later, there was a amateur scientist named Gregor Mendel. He really loved math and science, but he couldn't get into college. So he ended up becoming a monk at a monastery and he was a gardener. He really liked plants. And he ended up conducting all of these experiments on his own and taking meticulous notes, and really wasn't appreciated until after he died, because he was thought of as not very smart at the time. But in fact, did some groundbreaking work in genetics. So he was using pea plants, and pea plants were really great because they reproduce really quickly, and they happened to have really simple genetics, which was just very fortuitous of him. If he had chosen a different plant, he may not have discovered anything. So that means that peas, for a lot of pea traits, one gene equals one trait. For humans, we're a bit more complex, and there's very few genes or very few traits that are caused by one single gene. So anyway, he did a lot of crossbreeding of plants to look for the statistics. He basically performed a bunch of statistics on his results to show that there is some material, some kind of physical material that's passed from the parent to the offspring in a predictable pattern. All right. And that's, basically what he discovered. So there was still no concept of DNA, but since he was able to get, repeatable results in terms of his statistics, if you had a yellow pea and a green pea and you cross them, you would always get 2 yellow peas, always. And then if you took 2 of those yellow peas and you cross them, you would get 75% of the offspring would be yellow and 25% would be green every time. So that said that there was something being passed down through these generations in a very predictable and consistent manner. And so he said that these traits, did he coin the word gene? I don't think he did. But he but he coined this idea that a trait is coded for by a gene, and a gene is a heritable piece of information. But the chemical form of that information was still not understood. So then the quest became, and this was like another 70 years later, because like I said, his data was not well respected till posthumously, till after he died. So in the 1920s, people started to take this idea more seriously, that there's some, physical substance that's being passed on and that's contributing to different traits. So this experiment was done by Frederick Griffiths, and it was very famous. So what he did was he took some bacteria. These are streptococcal bacteria, the same ones that like cause strep throat, basically. And he had two strains. He had one strain that when they grew on a petri dish, they were very smooth looking. And so he called them S cells. And he had another strain that when they grew on a petri dish, they looked kind of rough and matte. So he called them R cells, smooth and rough. The smooth ones, when they were injected into a mouse, made the mouse very sick and the mouse died. But the rough ones, when injected into a mouse, the mouse didn't get sick and just, fought off the bacteria and didn't die. So he took those S cells and he heated them up to kill them and injected a bunch of the dead S cells into a mouse. And of course, since the cells are dead, they couldn't cause an infection and the mouse is fine. But then this really interesting thing happened where if he took those dead S cells, which don't kill a mouse, and those R cells, which don't kill a mouse, and he mixed them together and injected them, the mouse would get sick and die. And when he opened up the mouse to get a culture of the bacteria, he found live S cells. So those dead S cells essentially transformed the live R cells into S cells, transformed them into these killer types of bacteria. And so he started referring to this inheritance molecule as a transforming principle, that these cells were transformed, and so there's some kind of transforming molecule. But what is this transforming molecule? That's what we don't know. So the next test that was done was about 15 years later. What was that, about 16 years later? Avery, McLeod, and McCarty did an experiment to try to, they basically took those same bacteria, the S cells and the R cells, and did some more experiments. So they took those S cell bacteria, the S bacterial cells, and they basically chopped them up, blended them up, and separated them into DNA, RNA, protein, fats, and carbohydrates. So they chemically separated out the different chemicals of the cell. Proteins have a lot of functions, and everyone's money at the time was that proteins must be responsible for being the inheritance molecule. But they ran this very simple test to address this. So they took each of those tubes and they added an enzyme to destroy that material. So I should restate, they didn't separate it out. So they took that mixture, that blended mixture, and in one tube they put in an RNase. And RNase destroys all RNA. So all the RNA was destroyed. They mixed it with R cells, and they still got S and R. There was still transformation. So it wasn't the RNA that was the transforming principle. Then they took a tube of ground up dead S cells and they treated it with protease, which digests proteins. And they mixed it with R cells and they still transformed into S cells. So proteins were not the transforming principle. Then they also did the same thing with lipids and carbohydrates, and finally they did it with a DNase. So they added DNase, which chops up all the DNA in that solution. They mixed it with R cells, and that's the only time they did not get transformation. So what that told them was that the DNA was the molecule that was causing the R cells to transform into S cells. That DNA is the transforming molecule or the inheritance molecule. And that was a big surprise because people didn't think DNA was really all that. They just knew they were some nucleic acids found in the nucleus and didn't seem all that interesting. Well, once they realized that it was the inheritance molecule, then it became really interesting, and we wanted to know much more about it. One of the things that you need to know about a molecule in order just to study its function is what is its structure. And the structure of DNA wasn't known. And so there was a race, basically, to try to figure it out. And there were lots of independent researchers who did little studies that came up with important pieces of data that helped to solve the puzzle in the end. But the first one of note that was really important was Erwin Chargaff. And Chargaff took DNA from different organisms, and he basically just measured the amount of adenine, thymine, guanine, and cytosine in the DNA of different organisms. And he found different organisms had different proportions of each, but no matter what organism he looked at, they always had the same amount of adenine as thymine, and they had the same amount of guanine as cytosine. So just to like play with some fake numbers here, let's say they had 20% of their genome was adenine and 20% was thymine and 30% was guanine, 30% was cytosine. So they didn't necessarily have equal amounts of all four, but whatever amount they had of adenine, they had of thymine, whatever amount they had of guanine, they had of cytosine. So that was an interesting pattern, but he couldn't reconcile what it meant. He knew it was important, but he couldn't figure out what it meant. A year later, Rosalind Franklin and Maurice Wilkins, working in labs next door to each other, they were both crystallographers, so they worked on crystallizing molecules in order to basically take pictures of them with an X-ray. So it's called X-ray diffraction, and it's very tedious work to crystallize molecules, and DNA just happened to be very difficult to crystallize. But Rosalind Franklin happened to be very skilled and figured out how to get a really good clean crystal of DNA and got a really nice picture of it right here. And this picture here, although I don't know how to read an X-ray crystallograph, this shape here is very indicative of a double helix structure. So she was the first to really identify that the three-dimensional structure of DNA was a double helix, a twisted ladder. So her work with Maurice Wilkins was sort of, they were doing it on their own, but then these two guys came along, James Watson and Francis Crick, and they were young PhD students. They were just full of energy, really go get 'em, and they were fully invested in this race to figure out the structure of DNA. And so they met Erwin Chargaff at a conference and learned about his data. And they also met with Maurice Wilkins, who was talking about his colleague, Rosalind Franklin, who taken these great pictures. And he showed them the pictures, the X-ray crystallographs that showed it was a double helix. And so they took all of that information and kind of synthesized it, and a lot of hard work and also a little bit of good luck and determination, they were the ones that sort of solved the puzzle and figured out that yes, DNA is a double helix, but that they figured out how the nucleotides fit and are bonded in the latter rungs here. And they published a really elegant and simple, like one-page paper that detailed the structure of DNA and was the correct structure of DNA. So that's 1953, guys. 1953. That is like 68, 67 years ago. Not that long ago. Not that much. Not long before my mom was born. She was born in 56. And so 3 years before my mom was born, the structure of DNA was discovered. All right. And by the time I was a kid in the year 2000, the turn of millennium, we were already sequencing the human genome. So we went from discovering the molecule to being able to sequence the entire genome of an organism in like 50 years, which is just insanely fast in terms of progress. So the progress in molecular biology over the last century is just crazy. And now, I mean, it's 2020 now, not 2018, but you can pay 50 or $100 and spit in a tube and send it to a company, and they will sequence your entire genome and send you the sequence of your genome. Like, it's just crazy that we've had that much advance. It took the first human genome sequence took, I don't know, 10 years or something. It took several years just for one. And now it takes a couple of weeks for one person to just do it. So, hundreds of people can get it done at a time. So it's pretty crazy, the advances that we've had in this field and the greater understanding that we have now for genetics. So let's get back to that DNA structure. So we know that DNA is a double helix, so it's made up of two strands. The strands run anti-parallel, so one runs up north to south and the other runs south to north, or maybe this one's south to north and this one's north to south. In DNA language, we say 5' to 3', and the other one is 3' to 5'. So they're running in opposite directions, like a highway that goes north and south in opposite lanes. And then the rungs of the ladder are made up of nucleotide bases. Okay, so we call this the sequence of the DNA, the primary structure. Just like we did with polypeptides, the sequence of amino acids is the primary structure. And the secondary structure of the DNA is that double helix shape. Okay, so these rungs, these nucleotides that are bonded together, they are bonded based on complementary bond pair rules. So certain bases will only bond with certain other bases. And the bonds that hold them together are not really bonds. They are attractive forces that we call hydrogen bonds. Even though we call them bonds, they're really attractive forces. So the hydrogen bonds holding these two strands together are really quite genius, because the beauty of the hydrogen bond is that it's very strong. So it keeps your DNA stably in this structure, but it's also not as strong as a bond. It's still an attractive force, so it's also easy to open. To me, a good analogy is a zipper, right? So you zip something, it stays shut. Like, my jacket is not going to open. If I rip it like this, the zipper is sturdy. But when I need to open it, this little zip tag here, I can just very easily open that zipper. So that's kind of like how the DNA is structured. And that's important because it's your genetic material. It's the instructions for everything your cell needs to do. So you need to keep it safe and stable and intact. But when those instructions need to be read, you need to easily be able to access them. So being able to easily unzip the DNA to access those instructions, or to make copies of the DNA to make new cells, is relatively easy. So those complementary base pairing rules are that guanine always pairs with cytosine, and there are 3 hydrogen bonds that hold them together, and adenine always pairs with thymine, and there's 2 hydrogen bonds that hold them together. There's a channel on YouTube called Amoeba Sisters, and they have great biology videos, little short 5-minute cartoony videos. And they have a really good mnemonic for remembering these pairing rules. And it's apples in the tree, A binds with T, and car in the garage, so C pairs with G. So when you have 2 strands that are running anti-parallel to each other, they have sort of we say they have complementary sequences. So if we wanted to indicate the complementary strand of this little piece of DNA, first of all, we would have it running anti-parallel. So the ends would be switched. Instead of 5' to 3', this one would be going 3' to 5'. And the opposite of that adenine would be a thymine, apples in the tree. Opposite the cytosine would be a guanine, car in the garage. G complements C, A complements T, T complements A, C complements G, and T complements A. So the opposite strand of this piece of DNA here has the complementary sequence. So there might be problems in the homework or on a test that ask you to write a complementary sequence. So just like with proteins, we had primary, secondary, and tertiary structure. So the secondary structure is just the helix, but the tertiary structure is a more three-dimensional folded up form. And in DNA, what happens to our DNA, you actually have like 6 feet of DNA in each cell of your body. It's kind of insane to think about 6 feet of DNA all crammed into a nucleus of one cell. And the way that we do that is through supercoiling. So if you think of like taking a string, you could do this with your hair too. If you take a string and you twist it, twist it, twist it, twist it, and you just keep twisting it, it'll just start to coil in the center. It'll just wrap up on itself from all that tension that's built up. That's basically what we do with our DNA. We wrap it super tight. We actually wrap it around proteins called histones, and then those get even further coiled, and it's called supercoiling. And they coil up into these tight bundles called chromosomes. So chromosomes actually are not usually in this X shape. Just think of like half the X. So a chromosome in cells normally just looks like this here. But when a cell is replicating its DNA, it doubles it, and then you have the X-shaped chromosome. But normally our chromosomes are just like a single line of tightly packed DNA. So humans have 46 chromosomes in each cell, 23 from mom, 23 from dad. So they have 2 pairs, or 23 pairs, which equals a total of 46 chromosomes in each cell. So DNA is in the nucleus, and it's double-stranded. It's got that double helix look. We're also going to talk about RNA, the other nucleic acid. So DNA, some of the differences between them is that DNA is mostly double-stranded and RNA is mostly single-stranded. Another difference is in those bases. So DNA uses the 4 bases A, T, G, and C, and RNA uses the bases A, U, G, and C. So DNA uses thymine, where RNA uses uracil. It's something that you can use to differentiate between the 2 nucleic acids. They also have different sugars. So DNA has deoxyribose, whereas RNA has ribose. And they're found in different places in the cell. DNA is in the nucleus, and RNA is made in the nucleus, but then it goes to the cytoplasm. So a lot of it is found in the cytoplasm. So we said, I gave you that sort of analogy that, your DNA is kind of like a recipe book. And what meals you choose to make from that recipe book is, determined by lots of different things in your environment, I suppose. And so when a cell has all those instructions in the DNA in the nucleus. When it decides there's a gene that it wants to make, that needs to be made, all right, it makes a copy of that in RNA, and the copy goes out to the cell where it's turned into a protein. So it's kind of, I guess, if we're going with this cookbook analogy, which I just invented on the spot, it'd be like saying, if you have a very like rare cookbook, all right, you wouldn't want to bring it out in the kitchen and have it out on the countertop while you're mixing all your stuff, because you could spill food on it and you wouldn't want to ruin it. So you might want to take a picture with your phone or make a photocopy of the recipe and use that one to actually be your working copy so that you preserve the integrity of that very important cookbook. And so that's kind of how cells operate. The DNA stays in the nucleus so it can stay pristine. And the only, copy the original copy. And then, but when proteins need to be made, we just make these temporary photocopies of the genes using RNA. And the RNA is made in the nucleus. It's just a copy of a section of DNA. That process is called transcription. And then the RNA, that little RNA copy, is sent out into the cytoplasm, where it is read by ribosomes that make the protein that's coded for in that piece of RNA. And that process of going from RNA to protein is called translation. So this pathway in biology is often referred to as the central dogma of molecular biology, that all proteins in cells are made from RNA that's made from DNA. So DNA goes to RNA, goes to protein, and that first step, and this whole process of making proteins from the instructions in the DNA, is known as gene expression. And so gene expression has two parts. The first part is transcription, going from DNA to RNA, and the second part is translation, going from RNA to protein. It can be confusing because these two words look and sound a lot alike. They both start with trans and they both end with T-I-O-N. All right, but the way I remember it is transcription is to transcribe something. To transcribe something is to copy it. Like if you were listening to like a report on the news and you were typing it up for someone who couldn't hear and they could only read, you would be writing the transcript. All right, like if you go to a YouTube video, you can look at the transcript. All right, it's just a copy. It's saying the exact same thing in the same language. All right, just slightly different form, oral versus written. So DNA to RNA, DNA and RNA are both nucleic acids. It's the same, like we're just copying it from one to the other. Translation, though, is what you do when you're going from one language to another, right? So like if you're going from like English to French or Hebrew to Chinese, okay, then you're going from sometimes a language with totally different letters to another language with totally different letters and words. So that's kind of what's happening when we go from RNA to protein. We're taking something in a language of nucleic acids, and we are converting it into a new language, the language of proteins, into amino acid sequences. So when it comes to RNA, there's actually multiple types of RNA, but the three main types of RNA that we're going to focus on that are important for transcription and translation are listed here. So the first one is mRNA. The M stands for messenger. This is the one that was pictured in that last slide. So it's basically carrying the message from the nucleus to the cytoplasm. Ribosomal RNA is rRNA, and it makes up these protein reading or these protein making machines in the cell called ribosomes. So ribosomes are actually are structures made from both protein and RNA together. And then transfer RNAs, tRNAs, bind to amino acids and bring them to the ribosome. And so they help to make proteins. So ribosomes, all three of these actually are involved in translation. Messenger RNA is the only one that's involved in transcription. So transcription happens in the nucleus, right? So what happens there is the DNA gets unzipped. You can see in this picture, so the blue strands are those of the DNA. And then an enzyme comes in called RNA polymerase. Polymerase literally means an enzyme that makes polymers, and then RNA means that. So it's literally named an enzyme that makes polymers of RNA, and that's what it does. It comes in and it reads the DNA sequence and it generates a strand of RNA that is complementary to that strand of DNA. And so we can give each of these strands of DNA a name. We call this one on the bottom here the coding strand, because this is the one that actually codes for a gene. A gene is a segment of DNA that actually codes for a protein. So you could kind of think of it as like a recipe in that massive recipe book. Okay, so a single recipe. So we want to copy this recipe, and so we have to use the opposite strand as our template so that we can make an RNA strand that is identical to the coding strand, all right, to that gene. So the template strand is just the one that we use to make a copy of the one we actually want, which is the opposite strand. You'll notice that these RNA bases are complementary to the ones in the DNA, but wherever you would expect there to be a T, complementary to the A, wherever there's a T in the coding strand, there's a U in the RNA transcript. So remember, RNA uses uracil, whereas DNA uses thymine. So once that mRNA is complete, it leaves the nucleus and goes into the cytoplasm where it can encounter ribosomes in order to make proteins. A quick sample problem here from the textbook. The sequence of bases in a DNA template strand is CGATCA. What is the corresponding mRNA that's produced from this DNA? So this is the template strand, the one that we are going to be making a complementary sequence to. So if this is the DNA sequence, what would the complementary RNA sequence be? I want to change my pen color, but I don't know how. So the RNA sequence would be the complement. So G, C. Complementary to A is usually T, but since this is RNA, it's going to be U. Complement for T is A. Complement for C is G. And the complement for A is T, but since this is RNA, it will be U. Okay, so that is the corresponding RNA that's produced. So that's transcription. And what we just did in this slide that's already been erased is we did transcription. We took a DNA template strand and we figured out what the sequence of the RNA strand would be. The next step, once that RNA goes out of the nucleus into the cytoplasm, is it needs to get read by a ribosome. And ribosomes translate the code of the mRNA into the correct sequence of amino acids to make a protein. So I like to say the ribosome reads the mRNA. So here's the ribosome in red. It's actually it's like a hamburger, I think of, like two buns coming together. And then this yellow strand here represents the mRNA, the messenger RNA. And here's a tRNA in purple. And connected to it is actually a string of amino acids. So that this tRNA brought in this amino acid right here, which bonded to other amino acids that were already in process in this ribosome. So this is a very dynamic process, how the ribosome reads the mRNA, and they read it in little 3-letter segments called codons. And I feel like an animation is just a better way to really view and try to comprehend the process of translation. So we're going to watch a short animation here, probably after an ad.
My name is Scott Harrison. Just listen. This is my wife.
In prokaryotic cells, translation is initiated by formation of an initiation complex consisting of the 30S ribosomal subunit, formylmethionine tRNA, and messenger RNA.
All right, I don't like this guy's voice. It's annoying. It's a computer. So I'll just narrate. So we've got the ribosome. It's in 2 pieces. It comes together. This pink strand here is the RNA, and here you can see the RNA bases. This is a tRNA, and the tRNA is bound to an amino acid. And you can see that that tRNA also has 3 amino acids down here that are complementary to the 3 amino acids in the mRNA. So this, in the mRNA, that 3 amino acid sequence is called the codon. And I think that word will pop up here soon. It's called a codon. And on the tRNA, it has the complementary sequence called the anticodon. So that's how the tRNA, the correct tRNA, brings the correct amino acid. Whichever one has the right, the anticodon that matches the codon will bind there. And then when two amino acids are next to each other in the ribosome, a peptide bond forms. The ribosome shifts. Now that empty tRNA leaves, it goes to get another amino acid. And the next amino acid comes, or the next tRNA comes in, bringing the next amino acid that has the right anticodon to match the codon. A peptide bond forms between them, and this chain elongates, and the ribosome keeps moving. And this tRNA will leave through the exit site. And so this process just keeps going. The ribosome keeps moving down the RNA, and amino acids are added one codon at a time until it reaches the end of the gene sequence. And the last codon of the gene actually codes for a stop. It basically, kind of like a telegraph, like you write a sentence and then you say stop, like so-and-so had a baby, stop. Everyone's healthy, stop. Much love, stop. Like that's how a telegram would read. Okay, so that's kind of how protein sequences read as well. There's a stop code at the end that tells the ribosome to stop at that point. So the tRNAs, so the mRNAs have sequences of nucleotides, so they have A's, U's, G's, and C's, and they are read 3 letters at a time. So a 3-letter sequence on the mRNA is called a codon, just like you might say a 5-letter sequence of English the alphabet is a word. Okay, so a codon is like a word. Like when you're reading a page, you read it 1 word at a time. Okay, so each of these codons is read 1 at a time. And the tRNAs have an anticodon, basically a complementary sequence that matches the codon and always brings the same amino acid to that codon. And so the RNA code has been decrypted, and we figured out with all of the different nucleotide patterns, there's 64 different possible codons. So 64 possible combinations of 3 made from those 4 different letters. And they all code for either an amino acid or a stop signal. So we'll see that some, like serine, for example, okay, there's 4 different codons that code. Actually, there's 6 because there's 2 more over here. All right, but for some, like methionine, only has 1 single codon. And then there's 3 different stop codons. So these tRNAs, depending on which anticodon they have, they bind a specific amino acid and bring it in. This is just another animation that I'm not going to actually play. So if we were looking at the whole picture, transcription and translation. So you have DNA, and here is the template strand of your DNA. Here is the complementary mRNA strand that you made. So this is actually an mRNA version of the DNA gene, the coding strand. And we read these in triplicates, in little groups of 3. So codon 1 is UAC. And if we refer to this slide here, UAC right here codes for tyrosine. And hence we get tyrosine. GAG is a codon that codes for glutamate. UCG codes for serine, and so on and so forth. And that's how a protein is built and how it's coded for from originally the DNA. So we could do a little practice translation here. We could translate, we're given an mRNA sequence here, and we're going to read it and convert it, translate it to the amino acid sequence. So that's the mRNA sequence, and we're going to write out the amino acid sequence. So GUC is right here. That encodes the amino acid valine. AGC, where are you? Right here, encodes serine. And CCA is right here, and it encodes proline. So this mRNA encodes the amino acid sequence valine, serine, proline. I just like having lots of diagrams showing this process of the central dogma. And I really liked this image as well, so I just have a couple of them in here in the lecture notes for you. So this one's showing both of the strands of the DNA, and the coding strand and the template strand, so that you can see that the mRNA is complementary to the template strand, but it's identical to the coding strand, with the exception of replacing, T's with U's. And then we can see all these the tRNA anticodons that are complementary to those RNA codons, and we can see what amino acids they encode. So it can get confusing. I think sometimes the anticodons get confusing. So remember, it's the codons that we use to read that table to determine which amino acid is going to be made. So CUG codes for leucine, etc. So let's do a quick practice problem of transcription and translation ourselves. So the first thing we need to do, this is the DNA, and I'm meant for this to be the DNA template strand, so I'll specify that. The DNA template strand. So our mRNA is going to be A, U, G, 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 and I have them split up into 3 so that our codons are easier to read. This will be G, G, A. ATA will be U, A, U. And CGC becomes G, C, G. All right, I'm going to skip over because it's just sometimes confuses students. So we're not even going to worry about figuring out the anticodon. We're going to go straight to figuring out the amino acids that are coded by each of these. So AUG codes for methionine. All right, and GGG, I'm going to let you do the rest on your own, because if I flip back in my slide or try to leave this screen, I'm going to lose what I've written here. So you just have to use that genetic code to look up what amino acid is coded for by GGG, GGA, UAU, and GCG. So one of the things that you notice about the genetic code is that there are those stop codons, all right, and I didn't mention this, but there's also a start codon. AUG is sort of like a universal start, like it says this is where the sequence starts, and then there are 3 different stop codons that say this is where the sequence ends. And it's basically directions for the ribosome. So the ribosome is a big protein that docks on the mRNA. So you can think of the mRNA as like a landing strip, right? So think of a plane. Think of a landing strip for a plane, all right? The plane doesn't land like right at the edge of the landing strip. Like they start to come down, but they usually, they aren't aiming to touch down right at the edge of the landing strip, because what if they miss? And so they'll, if this is the edge of a landing strip for a plane, it might come in here, and then it has a lot of runway space to slow down before it comes to a stop. So I like to think of that for ribosomes, that there's sort of like this runway space, this extra space for the ribosome to bind and then kind of cruise along before it actually has to get to the place where it begins reading. So there's extra nucleotides on either end of the code of the mRNA, because the ribosome just needs space to dock and then needs space to kind of fall off after it's done reading. So that's why we need a start codon and a stop codon. You need it to tell the ribosome where to start and where to stop, because it needs a little bit of room to land first before it starts. Another image, again, just showing you that flow of gene expression. That first it's transcription, we go from DNA to RNA, and then we do translation, go from RNA to protein. So Another thing that can happen in genetics, of course, is mutations. So mutations occur at the DNA level. A mutation is any change in the DNA that's, different from normal. So some changes are harmless. Sometimes you get a change in the DNA and it doesn't change the amino acid. So remember, we had a lot of redundancy in the genetic code. So CAA and CAT in the DNA both code ultimately for valine. So changing this base, this DNA base from A to T, doesn't actually change the protein sequence. We call that a silent mutation. It's a mutation because it is a change in the DNA, but it's silent because it has no effect ultimately on the protein. Another type of silent mutation is one where you change the base and it does change the amino acid, but it changes to another very similar amino acid. So valine and leucine are very similar. They're both small non-polar amino acids, so they have very similar chemical properties. So it's almost like a not noticeable change in the protein sequence. So that also might be considered a silent mutation. All right. You also may think, oh, a single mutation, a single point mutation at one point and one letter of the DNA changing, that's probably not a big deal. But it can be a big deal. One base change can change a protein so drastically that it leads to a systemic disease. And the classic example of this is sickle cell anemia. So sickle cell anemia is a disease it's due to a mutation in the hemoglobin protein, which is basically the main component of your red blood cells. It binds oxygen and it's a globular protein and it helps give your red blood cells their typical donut-looking shape. Well, when you have the sickle cell mutation, it's a single point mutation, so this T in the DNA sequence becomes an A, all right, and it ended up changing the amino acid sequence from a glutamic acid, which is an acidic amino acid, so it has a negative charge, it's a negatively charged amino acid side chain, to a valine, which is a nonpolar amino acid, so they have very different chemical properties, very different attractive forces, and they end up causing that one change in that one amino acid ends up causing the protein, the hemoglobin protein, to fold improperly. And instead of forming a globular structure, it forms something more elongated, and they have a tendency to stick together, all the hemoglobins. And so they end up forming these long bundles, which stretch out the red blood cells into these elongated shapes. And so this is called sickle cell anemia because the cells look sickle-shaped, and a sickle is like a crescent-shaped cutting device. Okay, and those sickled cells, because they're elongated, they tend to get stuck in the capillaries, and they can cause blockage of blood flow. They can lead to strokes or heart attacks, definitely peripheral pain in capillaries where they get stuck. So they don't bind oxygen as well, so they lead to the anemia part of the disease. All right, so a single change in a single DNA base can change a single amino acid that changes a single protein found in a single type of cell of the body and still can be deadly. Is kind of crazy how, it's a snowball effect. When you have one little mutation, the effect of it can snowball as you realize how important that one amino acid actually is. So some different types of mutations. You can have mutations where there's a substitution, and that's what we were talking about with sickle cell anemia, where one of the bases is changed. So in the normal DNA sequence, it was CG, and here it got changed to T and A, and that ended up causing a change in that one amino acid there. All right. The more devastating types of mutations tend to be ones where you're adding or removing a nucleotide. When you do that, you end up shifting the whole reading frame. So notice when you when you delete a base here, or if you insert a base here, not only is that one amino acid right there altered, but all the amino acids after that are going to be altered because the reading frame shifts. It changes all of the codons after that. So my little like analogy or example is if we had this sentence that's all 3-letter words, the fat cat ate the rat. If we deleted that C and shifted everything, so they're still 3-letter words, it would then read the fat Ada tet her at, which doesn't make any sense. Now the sentence doesn't make any sense. So those are called frame shift mutations, ones that cause a shift in the reading frame and affect all of the amino acids after that point. So how do we get mutations? How does DNA get mutated? Well, sometimes it's spontaneous. When your cells are dividing, they make copies of the DNA. So the DNA gets unzipped and there are enzymes that go on through and they make copies. Well, you know, they're not perfect. They're very good editors and they make very few typos, but occasionally a typo slips in. That's, you know, we all are prone to that, you know, when you're typing something and you accidentally have a typo. So our DNA polymerase also makes typos, and it sometimes catches them and corrects them, but sometimes it doesn't. So every time your cells divide, you risk a mutation or you accumulate mutations. That's why some diseases like cancer is due to accumulation of mutations within a cell. That's why a lot of cancers become more common as you age, because the older you are, the more cell divisions you've had, and essentially the more typos you've accumulated in your cells over time. So that can happen randomly in cells as they form. It can also happen randomly in a developing fetus as those cells are developing. So some types of birth defects are inheritable. You inherit them from a family that had that mutation, or it just spontaneously arises due to a typo during fetal development. There's also substances that can induce DNA damage, that cause DNA damage, things that we call them mutagens or carcinogens because they contribute and lead to cancer. Different chemicals, like a lot of the chemicals found in cigarette smoke, are mutagens. Radiation is a mutagen, so X-rays are mutagenic. They damage DNA, so you have to be careful about your exposure to radiation. There's also a difference between somatic mutation and germline mutation. So somatic mutation is when you get a mutation in one of your body cells, not your egg or sperm cell. So if you had a mutation in a breast cell, and if you had several mutations there, you might end up with breast cancer, or a skin cell becomes very mutated from overexposure to the sun, from radiation. All right, it may develop cancer. But if you develop skin cancer, you can't pass that on to your child. It's not heritable, and it is not contagious. All right. Cancers, one way that cancers are inheritable, though, somatic cancers are inheritable, is that, so on average, a cancer cell has about 20 mutations. So if you genetically are born with 15 mutations, you only need 5 more mutations to develop cancer. So your risk of developing cancer is higher. You may have a lot of family members who have developed different types of cancers. So cancer can run in families. Really what it is, it's a predisposition to cancer that can run in families. There are certain types of cancer where there are certain genes that are more likely to result in cancer. But otherwise it's not directly heritable. The type of mutation that is directly heritable are ones that occur in the germline. So if you had a mutation that occurred in an egg cell or a sperm cell, then that's going to carry on to all the cells of your offspring. And that's where you get genetic diseases. So like sickle cell anemia, for example. All right, so a little concept check problem from the textbook. An mRNA has the sequence of codons CCC, AGA, GCC. If a base substitution in the DNA changes the mRNA codon of AGA to GGA, how is the amino acid sequence affected in the resulting protein? So I need to do this one. So I'm going to look up a genetic code, pull it up on my phone so I don't have to go away from the screen. And here we go. Okay, so CCC is proline. I don't like this table. CCC is proline. AGA is arginine. And GCC is alanine. All right. And in this problem, it says if there's a substitution in the DNA and it changes the AGA codon to GGA, so if we were to change this to GGA, how is the amino acid sequence affected? So AGA is arginine, but GGA is glycine. All right, that's a problem. So arginine is, if I can remember, I think arginine is a basic amino acid, so it has a positive charge. It's polar and it's positively charged. Glycine is a nonpolar amino acid. Right, so these two amino acids have very different chemistries. Nonpolar and polar charged are two very different types of things. So do I think this will have an effect on the protein function? Yes, I do. I think that I would predict that this would have an effect on the protein function because they have very different chemistries, different attractions that will affect the three-dimensional structure, because this arginine might be involved in a salt bridge that holds the 3D structure together, and now it can't do that salt bridge because we've put a glycine there. So yes, I would predict that it would have an effect on the protein function. Oh, and that's the end. I forgot that I didn't get into viruses in this chapter. So the chapter does go into viruses a little bit, which I probably should get into since we're in the middle of a pandemic with a virus. But I feel like just getting a basic grasp of nucleic acids and coding and how that works is a good start for now. And then you can come take microbiology with me and I'll tell you all about the viruses.